Good morning, everybody. So welcome to our second lesson on life sciences in grade 12. And today we are going to be looking at the following. We are going to be looking at DNA, RNA, and inheritance. We are going to be doing all the exam questions on those topics that is found in the 2020 paper 1 and 2 of the national exam that happened last year. So topics that we will cover today includes DNA versus RNA, the differences, what they fundamentally are, how we understand them, DNA fingerprinting, and how to answer questions involving that, some genetic crosses, and then family tree diagrams. So starting off with DNA versus RNA. So life on Earth is very diverse, and what DNA and RNA is the fundamental building blocks. Think of it as the Lego of life. So the basic Lego that you build the walls out of is the DNA molecules, and then the little frilly parts and the windows and the connectors of the RNA. Let's look at the basic differences between DNA and RNA. This could always be a nice question to ask in the exam about the differences, or to tabulate the differences. Maybe remember, when tabulating, you must draw a nice enclosed table with table headings. So if we look here and we look at the differences between DNA. So DNA, starting off with that, it is called deoxyribonucleic acid, and it is a double strand. You can see that DNA is in the form of, we call it a helix, so it's making these twists, and it's a double helix. It's two intertwined twists. It is also a polymer, and if we think back to grade 10, we learned that polymers are long molecules that are made up of little small molecules built together. So they're basically a long chain of small individual molecules bound together through a distinct bond. Excuse me. So this DNA, the first difference we can see is it is a double helix, where if we look at RNA, we can see that it's only a single helix. There's our first difference. Our second difference in DNA is the nucleic bases. So we can see DNA is primarily made up of these four bases: thymine, cytosine, adenine, and guanine, where RNA is built up of these four bases: uracil, cytosine, adenine, guanine. Spot the difference. RNA molecule has uracil and a DNA molecule has thymine. So if you ever get asked a question, and maybe especially in the protein synthesis questions, you're confused with what molecule you are working with, the first thing that should give it away is search if you can see a T or a U in those bases in the protein synthesis question. If you see the T, then you know you are working with DNA, and therefore we can know that we are probably working with DNA replication. But if you see a U in the question, it's going to be something to do with protein synthesis, maybe transcription or translation. So there's a nice first trick to know which molecule you are working with if you're a bit confused. Back to some of the other differences. The third difference is where these guys are located. So we find DNA in the nucleus. We think back to grade 10 and even did that in grade 9, where we looked at the cell and all its organelles. We learned that the nucleus of a cell, we used to say a metaphor, it is the brain of a cell. So that's where the DNA is found. That is where the basic instructions for everything else is located. where RNA is found in the cytoplasm, nucleus, and ribosome. So cytoplasm, there's some in the nucleus, and you will find it by the ribosomes. Why do we say that? If we just quickly take a break and we think about protein synthesis, oh gosh, sorry. Then with protein synthesis, remember, we want to make a protein molecule out of this DNA molecule. The DNA molecule is found in the nucleus. It unwinds. We make a little cute mRNA molecule. The mRNA molecule is our messenger. It takes the information out of the nucleus and it takes it to our ribosomes, where a protein is eventually made by combining lots of amino acids. So why we say we find RNA in all those places is there's free RNA drifting as tRNA in the cytoplasm. There's RNA we find on the ribosomes. And we find RNA drifting in the nucleus that's going to eventually bind together to make that mRNA to take it out. So that is why we find the RNA in all of those places. Our fourth difference between these two guys is we can see that the sugar molecule of DNA, if we say out the whole DNA word, it is a deoxyribose. So that first part of DNA is telling us it's a ribose sugar, but it's deoxy, it's got one less oxygen, where the RNA is just a ribose sugar. Another difference we can find is the primary function of these two molecules. So the function of DNA is the transmission of genetic information, and in a way, a long-term storage of information, where RNA is necessary and is used to transfer that genetic code into a protein and then eventually into all organs and functions in your body. DNA, just how we get more DNA. Remember that we learned about DNA replication. So it basically copies itself, where RNA doesn't copy on its own. It is made from DNA. So there's instructions in our DNA to make the RNA molecules. And then lastly, I just want to point out, when we said the differences between DNA and RNA is our thymine and our guanine, remember that all four of these molecules in DNA bind together in a set pair. We always have guanine and cytosine binding together, and we always have adenine and thymine binding together. So you need to remember that, where if we look at the RNA molecule, it follows the same rules, but instead of adenine and thymine binding together here, it's adenine and uracil, and then in guanine and cytosine is exactly the same in RNA and in DNA binding together. So those are our main differences between RNA and DNA. So the first question with that as groundwork knowledge that we find in the exam is question 2.1. So it reads, detectives were investigating a crime scene and found blood on a broken window. They suspected that the blood was that of a criminal. To identify the criminal, they analyzed the DNA sample from the blood and compared it to that of all four suspects. The diagram below was produced. So it is important to remember that DNA fingerprinting. So DNA, this question is about DNA fingerprinting. And don't get confused between the police taking DNA fingerprints, so doing the technique of DNA fingerprinting, also called DNA profiling, and taking normal fingerprints from a crime scene. So there's basically two types of fingerprints that police take from a crime scene. The first is the normal fingerprints that if you just touch anything, you can see it most clearly when you touch a car window, the oil on your skin, you can see the nice lines of your fingerprints left behind on that window. The other one is the one that we're focusing on, the DNA fingerprinting. So it's not actual fingerprints, it is pieces of genes, pieces of DNA that we are analyzing, that the scientists are analyzing, and they are comparing the genetics, they are comparing those nucleotides. How do they correspond with the crime scene and then with the killer? So they're looking how closely related the suspect's DNA is to that DNA they found at the crime scene. And remember, how they can get a DNA fingerprint sample is from skin cells, from hair cells, from blood, anything that contains some DNA in it can be used to take a DNA profile or to do DNA fingerprinting. It doesn't have to be a physical fingerprint, two different things. So if we look at this diagram below, on the far left, we see the DNA sample and then the four possible suspects, Bato, David, Jenny, and Tapi. 2.1.1, name the technique that was used to identify the criminal. So there I already gave you the answer. It is DNA profiling or DNA fingerprinting. Now, 2.1.2, who is the possible criminal? So how do we work that out? So here's our DNA sample. So this is the main block that we need to start when we look at anything. And it's basically now playing a game of finding the differences between two pictures. We are looking for the person that has most DNA bands. Each one of these is called a DNA band. And we want to see which one is most closely related to the sample we found at the crime scene. So if we just compare the first row, everybody still looks good. Everybody still has the same DNA sample. Comparing the second row, we can see, okay, David is not really close. Comparing the third row, David and Jenny. This row, Jenny is already corresponding a lot, almost all the time. And if we go down, Jenny looks like the perfect candidate because almost all the bands actually are exactly the same as in the one we found at the crime scene. So there we can say Jenny is our possible criminal. 2.1.3, explain your answer to question 2.1.2. So here it is very important that you always use one of the two words. You should use the word profile, or you can use the word band when you want to describe the suspect and the original DNA sample. So when answering these questions, make sure that you've got profile or band in your answer and also refer to the DNA sample and refer to your answer. So whoever you said was the guilty criminal or who looks like they are guilty, make sure that you mention all of those. So here the marks were awarded for saying Jenny's DNA profile, or you could have said Jenny's DNA band matches the DNA band or matches the DNA profile from the sample. See, we used all three of those words in our answer. So Jenny's DNA band matches DNA band from the sample. 2.1.4, state one other use of a technique identified in question 2.1.1. So the technique we identified is DNA profiling or DNA fingerprinting. Now here's quite a few techniques that we can use. Remember my tip in lesson 1 on answering questions. Make sure you look at the mark allocation. Do not waste your time giving more than one answer when the mark allocation is only one mark. And remember that markers only mark if you are asked to list something, they are asked to list 3 things and you list 6 things, they only mark the first 3 things. So make sure that you are not wasting time writing extra things that would firstly not get marked, and secondly, just waste your time where you could use them on other questions that you are struggling more with or are just more time intensive. So here, state one of the techniques one of the uses this technique can be used for. It can be used for proof of paternity. We can trace missing persons, identification of genetic disorders, establishing family relations, matching tissue for organ transplants, identifying dead persons or dead animals. Any one of those. There are some other examples how we can use this as well, so don't let that put you in a box, but there are some nice uses over there for DNA profiling. Okay, moving on to our next question, if my tablet allows me to do that. There we go. Sorry about that. Okay, here we start now talking about inheritance. So the question 1.1.6 read, a homozygous purple plant, big capital P, is crossed with a pink flowering plant, small letter p, to produce the F1 generation. One of the F1 plants is crossed with a pink flowering parent to produce the F2 generation. Now they ask us, which one of the following is the correct phenotypic ratio of the F2 generation? So to be able to answer this question, there's just a few terms that we need to be familiar with. Let's remind ourselves of everything, all the definitions of all of them. So firstly, homozygous. The prefix homo always means the same. And here we are talking about homozygous. We are talking about 2 of the same or identical alleles. So we know if we see anything that's homozygous, let's just remind ourselves it could mean capital P, or small letter p, as long as they are identical. The opposite of homozygous, if we think, is heterozygous. And that would refer to the 2 alleles are different. And that means it will look like this, capital P, small letter p. Next, they tell us, very important, they're telling us what it looks like. They're telling us it is purple. That means they are telling us the phenotype. Phenotype, think phenotype is physical. They're telling us the appearance or whatever is expressed physically. So if it is P, capital P, they are telling us it is purple. Next, they say it is crossed with a pink flowering plant, small letter p, to produce the F1 generation. So now they're giving us another phenotype. They're telling us it's pink and it's a small p. So we already learned and here is the trick to this question, that if something is dominant, it will express as purple, because there's one capital letter and that indicates dominant. And remember, if something is a dominant allele, if it's a capital letter, it is the one that will be expressed. The only way a recessive allele can be expressed is if there are two small letters, two of the same recessive alleles. That means this indicates to us that there are two small letter P's together that will give us pink as a phenotype. Cool, that was the first sentence. And they said that it produces F1 generation. So F1 generation means it's the first generation of offspring, the first children. So these two guys were crossed. Firstly, let's write it over here. And they said for one parent that we now know is capital P was crossed with two small letter P's to give us our first generation, and it would look something like this. Remember, this is, I'm now doing a very quick Punnett square, and this would mean that we just move down all of the letters and match them up. So first one I place in the block, and I place this one in the block as well. I do it the same with this. I placed it there, I place this one there, I place that one there, that one there, that one there, and that one there. Then we match them up from the two parents. Okay, so sorry for now making that all squiggled up, but can you see that all of the children came out for the F1 generation, capital P, small letter p. And what will this phenotype look like? It will be purple. How do we know that? Because they indicated that in the first sentence. We needed to deduce from the first sentence that this is a capital letter, meaning it will be the dominant allele, and the phenotype with the capital letter is purple. So if there's a dominant allele, it will show as purple. Now they go on in this question. This is all the info we just got from the first sentence. Second sentence tells us one of the F1 plants is crossed with a pink flowering plant parent to produce the F2 generation. So it doesn't matter, they tell us one of them, one of these ones is crossed. It doesn't matter which one, because all of them gave us big P, small p, is now crossed with a parent that is pink. And we know that a parent that is pink is the small p. So let's draw out quickly that Punnett square as well. So the one parent looks like this. It's a capital letter, small letter, and two small letters. Let's again take the capital letter down, take one there, take that one down, across, capital letter down, across. Okay, that is our F2 generation now. So now they are asking to give the correct phenotypic ratio. So the correct, oh gosh, phenotypic ratio. What can we see here? These two give us a capital letter in the combination, meaning immediately it is purple. So 2 out of the 4 is purple. And here we see 2 small letters, so 2 recessive alleles, meaning only recessive gives us 2 pink. And we can just simplify them to 1 purple, 1 pink. And there we go. 1.1.6's answer is A. And that was worth 2 marks. Now, this is quite a lot of thinking and seemingly a little innocent question for only 2 marks. But this is all of the information that you had to go through and process to figure out which is the correct phenotypic ratio for the F2 generation. So remember all the terms that we went through. If we quickly want to just go through them again, is we said We looked at the first sentence. It was a homozygous purple flowering plant, capital P. That was our first little hint that we are looking at the dominant allele. It will show if it's 1 or 2 because it's dominant, and it will show as purple. Crossed with a pink flowering plant, it's recessive. It is meaning that it needs to have 2 recessive, 2 small letters to be showing that it is pink to produce F1 generation. F1 generation means the first set of offspring from a cross. Then they said one of the plants from F1 generation, luckily they all looked the same, was crossed with a pink flowering parent, which we know must look like a small letter P, and it produces the F2 generation, and they are asking us for the phenotypic ratio. And that was this question. Going on to question 1.5. This is inheritance as well, a Punnett square, and it reads, there is variation in the characteristics of fur color and fur texture in cats. The table below shows the alleles that control these 2 characteristics. So here we can see that we are working with 2 characteristics. So we are working with a dihybrid cross. So we're crossing 2 things at the same time. So it's not as simple as the previous question. Now we have 2 alleles. Let's just remind ourselves what an allele is. So an allele is one of 2 or more forms of a gene variant. And a gene, just remember, it is the basic unit of heredity that we work with, and it is made up of DNA sequences. So the characteristics we are going to be looking at here is the fur color and the fur texture. So let's look at the alleles. We can see that if it is the dominant allele, it's a capital B, gives us black. If it's a recessive allele, and remember for recessive, you're going to need 2 of these to give us a white fur color. For the fur texture, you can have 1 or 2 capital R's for the rough texture. And for the small letter R, that is our recessive allele, again, you're going to need 2 of the same for the smooth. And remember the terms that we know now is homozygous, meaning they will look exactly the same. So it could be RR or RR, but it means they are the same. Where heterozygous means that they look different. Okay. The Punnett square below shows the inheritance of alleles in a genetic cross, and there we see all the possible gametes from the female is over here on the left, and then all possible gametes from the male. Question 1 is they are asking us dominant phenotype for fur color. Remember, we can see that by the capital indication, and we're just working with fur color, so we must just look at the first block, and that dominant phenotype is black fur. Recessive phenotype for fur texture. Now they're looking at the second block. They just want to know the first fur texture, the recessive, and that is always the small letter, and that is smooth. Okay. Question 1.5.2 is asking us, give the genotype of offspring X. So where's offspring X? Over here. And remember, genotype is the genes. So here we need to give the alleles. If they're asking for phenotype, they don't want the alleles, they want the physical appearance. So firstly, A is asking, give a genotype of offspring X. How are we going to get that genotype? It's going to be exactly the same as we did the previous Punnett square. So we're going to take the gametes from the male and from the female, and we're going to be putting them together. So let's just write them out here. This is from the male, and simplified, this is from the female. So they're just going to be together, going to both move in there, because remember, they would combine. So you have two haploid cells combining to form a diploid zygote. So both of these cells, so both of these sex cells, remember that's what gametes mean. It is a sex cell and it is haploid. And haploid means half, and they're going to become one diploid cell if they combine. So here we have small letter b, R, R. Just arrange them to have the alleles that fit each other stand next to each other. So you can see they always write the B's first. So just follow the trend and write both B's first and then both R's first. Don't keep them combined. B says the phenotype of the female parent. So here we just have to look at the gametes from the female parent. So it's over here. Oh, my tablet is not working with me right now. Sorry, let me just erase all of these nonsense. Okay, so again, they asked the phenotype of the parent for the female, and that would be white fur with a rough texture. And lastly, C asks us the genotype of the male parent. So again, they're asking for genes, what it looks like. And the genotype for the male parent would then be, let's look at it. The male parent is sending a big B, a small b, a big R, and a small r. How we know that is we take all of the possible things that he is sending. If we look at what they gave us here a big R. If we look at the gametes, they gave us the big R, and the big B here in the second one, small r, small b, big R, and big small b and small r. Put that all together, all of the variations means he's actually sending one of each. Lastly, it is saying 1.5.3 says, state the phenotype that all the offspring of his genetic cross have in common. So if we look at all the offspring, so it is this last little row here that you would see at the top of your screen, we can see that something they all have in common, and it should be a phenotype. So they're all expressing the same rough texture of fur. So let me just show you how to get there. So this is all of our offspring at the top here. And they want you to show the phenotype, so only the physical appearance. So what is this first offspring showing that we can see here? We can see that it is showing the big B is small b. That would indicate, remember, big B, small b shows us the color. It has a big B, so it is automatically it has black fur color, and two big R's, meaning that it has rough fur texture. Big B, small b, big R, small r for the next rat means it's also showing black and it's also showing rough. The next one, X, is we saw that it was small b, big R, big B, big R, and that means it has a white fur, and that immediately disqualifies it for the phenotype. So it can't be white that everybody is showing, and it can't be black that everybody is showing, because we've already got 2 black mice and now 1 white mice. But again, it's showing big R for rough. And lastly, we can see this one has got a big R and a small r. If it's got a big R, it's meaning it's showing rough fur texture. So our answer at the bottom is rough fur texture. Okay, and that was this question. I hope that made sense. 2.3. So this is again inheritance, and now this is inheritance of blood groups. So what do we know about blood groups already? If we just think back what we learned, we've got an A, a B, and an O blood group. So this question reads, a man with blood group AB, first thing they tell us, and a woman with a heterozygous blood group B. Very important, they're giving us another clue. B. Plan to have children. So 2.3.1, how many alleles control the inheritance of blood groups? So I've already just given you the answer. There are 3 types of alleles that control blood groups. Next one, they ask, describe the type of dominance that occurs in the inheritance of blood group B in the women. The types of dominance that we have learned about is complete dominance or incomplete. So what that means is, I kept telling you now with that mouse fur color, as soon as it has a capital letter, it means that it only expresses the dominant allele. But the only example that we learn about that has incomplete dominance is in blood groups. And the nice example we find in blood groups is blood group AB, because A and B are 2 capital letters, and they are both basically, think of it as both dominant alleles, but you express them in this example at the same time. But it is not asking AB, it's asking for B. So that means this woman has B and something else over there for her blood group. Now the question is it BB or is it BO? And that would obviously mean if it's complete or incomplete dominance. But they told us in the question, this woman is heterozygous. So that means it is different. So that would mean her blood group needs to be BO. And we don't write the O over there, we just write the B. And that would mean that her blood group is B. And that would mean it is complete dominance. So the type of dominance they're asking here, the answer is complete dominance. Why? Because the allele for blood group B is dominant and the allele for blood group O is recessive. So remember, just from the theory that you've learned, A is dominant, B is dominant, and O is recessive. Last question they are asking us is, use a genetic cross to show the possible genotypes and phenotypes of the children. And this is 6 marks. So it is important to understand the basics of a genetic cross and where you will be getting your marks when writing a genetic cross. So first off, we need to start with the parents. So let me just erase all of this. Hopefully there's nothing here. Okay, parents. The parents we can see. Why is my tablet doing this? Okay, there we go. Parents. So we have learned that the father is AB, and he is going to be crossing with the mother, which is blood group B. How we write that in the genotype is the father will be Can I just ask if anybody can unmute and just tell me if your words is also disappearing on the screen as I write, or is it just on my tablet?
Ma'am, it's also disappearing.
I'm so sorry. I have no idea why. Oh, that one's staying there. That's good news. Okay, so hopefully it stays there now. So I just wrote P1 for the parents. The phenotype is blood group AB for the dad, which would cross with blood group B for the mom. The genotype is, remember, we need to write I's, so that's IA, IB crossing for the dad times I for the mom. Sorry, those are capital I's. Please just remember that. IB for the mom, and then a small letter I indicating our blood group O. Now we can do a genetic cross. There are two ways. The way I find the easiest, you get both marks for either way, so don't worry. It's just drawing the plain Punnett diagram like this, little block. And it doesn't matter where the dad or the mom is. You can just draw it there. IB and small letter I. And then you match them up saying IA, IB, 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 IA, I small letter, IB, small letter. So you get a mark for the correct gametes, meaning the correct things that you wrote inside that block. So 1 mark for this. You get a mark for the correct genotypes. And then you will get marks for giving me the genotypes of the parents over here. And you get marks, very important, for indicating P1, F1, which is our children. This is our children over here, F1, P1. And very, very important, you need to write these two words always, meiosis and fertilization. So there's where your 6 marks is coming from writing meiosis, fertilization. Sometimes they give it at the top. So as soon as you have written P1 parents, you have given me the phenotypes, the genotypes of the parents. Those are 4 marks. Then the gametes crossing, you get your other 2 marks, and that gives you the total of 6. So they just wanted the genetic cross to show the possible genotypes and phenotypes. So I'm so sorry it's not showing. I will fix it. The rest of it was showing in the previous slide, which is very weird. It's just this one slide. Okay. And then lastly, we are going to quickly look at 2.5, which is this little family pedigree or family diagram. So it reads, Gold syndrome is a sex-linked genetic disorder. It is caused by a dominant allele XG. The diagram below shows the inheritance of Gold syndrome in a family. 2.5.1, name the type of diagram shown, and this I have already given away. It is a pedigree diagram or a family pedigree. 2.5.2a. Females, they're asking how many females are in this family. Now, this is easy. We always know females are always indicated by a circle. So you are just going to count all the circles and don't get tricked. They didn't give circles over here, but they have indicated that there are 2 females. So there are altogether 6 females. 2.5.2b says males in the F1 generation have Galt's syndrome. So how many of them? So F1 generation, remember that's our first offspring generation. So we're only looking at the first row of offspring. And how many males are there that are infected, meaning they are colored in, and there is just 1. 2.5.3, give Gabby's genotype. So Gabby here at the top, you will see she is the mother, the parental generation, and her genotype, so her genes. We don't want a physical characteristics. So what would it look like? It is sex-linked, so we need to give something with the X. And it is caused by a dominant allele. So meaning a female only needs to have 1 X with the disease to be able to show it. So how do we know what her genotype is? Is it an X with 2 big G, so X big G, or X big G, X small g? Now we figure that out by looking at what children she will have. So if she has children that are both affected and unaffected, it is safe to say that her genotype is X big G, X small g. 2.5.4, Anju and Pelusa have 4 children. Give a phenotype of their sons. So here we see Anju and Pelusa, and here are their children. And unfortunately, my silly screen is still showing the previous. Oh, maybe I can just erase it. No, it doesn't let me. Okay, hopefully this has now helped. Okay, so it's asking for Anishu and Pelusa, Anju and Pelusa, and their 4 children, but they've only told us there's 2 males and 2 females. But they just want the phenotype of their son. So how do we work that out? So we know already that it is caused by X with a capital G, and we can safely guess that Pelusa, his phenotype has to be X big GY, because he only has 1 X, because he's a male, the other one is a Y, and he has the disease, so it has to be the X big G. And Anju, how do we know what hers is? She is an unaffected female, meaning she cannot have 1 X that has a dominant allele. So it's 2 recessive alleles will make her unaffected, because we read in the question it is a dominant disease from a dominant allele. So we already know what the parents look like, and we can do a normal family cross. But remember, now you're doing one with sex linked, so you would do this X big G Y. You would take the mom small g X small g, and you will put it together normally, just like in the previous Punnett squares. Just remember that now, do not forget about the X. I see a lot of my students forget about writing the X, and they just write the small letters as in capital letters. You cannot write that. You have to indicate that it is a sex-linked disorder. So our last one has now gone away. But if you write that out, the answer is that how many sons will they have? All of their sons will be unaffected. So all of their sons will be healthy. And then lastly, I just want to tell you why you can know that without even drawing the Punnett square. Remember that the dad either sends an X or a Y to the children. And that means the dad has definitely sent the Y to the males because they are males. A mom cannot send a Y. A mom only has one of two X's to send to the offspring in the gametes. That means the X chromosome that the males will possess will definitely come from the mom. And the mom has two X chromosomes that are healthy. So they will definitely receive a healthy X chromosome, meaning that there's a definite chance that the children will not have the disease. So that's the explanation in words, but you can also draw it out for you in a quick Punnett square in pencil. And then lastly, 2.5.5, explain your answer to question 2.5.4 for 4 marks. So how do you explain this? Remember, they are asking you to explain, so they're not asking you to draw or give a Punnett square. You need to use words now. So you can use the parents' names. So you can either say Pelusa and Anju, or you could say mother and father. But let me read out what they wanted. They want you to say Pelusa is affected, or you could have just given the genotype of Pelusa, and Anju is unaffected with her genotype. The males will inherit the Y chromosome from the father or from Pelusa, and they would inherit an X chromosome from Anju. And those X chromosomes inherited are unaffected. And there's where you would get your 4 marks from. Okay, thank you. Are there any questions? You're also welcome to email me on this email address, which you can see if you have any questions regarding inheritance or DNA or anything we did in this lesson. Okay, I take that there are no questions. I hope that you have an amazing Saturday and good luck for studying. I'll see you next week.
Thank you, ma'am. Bye.
Bye.